All right, so the first video for today has to do with the basics of viral structure. Okay, so remember that viruses are acellular infectious agents, so they're lower than the level of a cell. All viruses are completely dependent on a host cell in order to be able to replicate. Okay, so in our very simplest form, the virus has to have two parts to it. It has to have some type of genetic information. And if you look in your notes, you can see that this genetic information can be quite diverse. There's at least 7 different types of genetic information that we can have in viruses. Everything from double-stranded DNA to positive single-stranded RNA to the spectacularly gapped double-stranded DNA viruses. Okay, but regardless of what kind, we have to have genetic information. The other thing they have to have is they have to have that genetic information protected. And so this is typically done with something called a protein coat that in terms of viral biology we refer to as a capsid. And so our very simplest viruses are the ones that we call naked viruses. And these guys are essentially nothing other than what we call a nucleocapsid. And so you can sort of see in the name, we're going to have nucleic acids, we're going to have that genetic information, and then we're going to have that protein coat, we're going to have the capsid covering it. And so the most simple virus that we have would have some type of genetic information surrounded by a protein coat. Okay? Now, viruses can be more complex than this, and so usually what we mean by that is that rather than being a naked virus, we would be what's called an enveloped virus. And so what this is this virus now has a membrane outside of the nucleocapsid. The membrane, often at least portions of the membrane, will come from the host cell, but this is an extra layer. Now, one part of the membrane that always comes directly from the virus, that's encoded for by the viral genome, are the spike proteins. And these are proteins that actually stick out of the surface of the envelope. And in terms of the virus, they have a number of potential functions. So those are in your notes, but for example, they can be involved in attachment to host cells, or they can be involved in certain types of enzymatic activity. What's really interesting from our perspective for these spike proteins is that they tend to have very good antigenic potential. So they tend to be parts of the virus that the immune system recognizes and that can lead to antibody formation. So if you're looking at the news surrounding COVID-19 at all right now, there's a lot of discussion about some vaccines that they're trying in people. A lot of those vaccines are using the spike protein from that virus to try to stimulate an immune response.